Hello students, welcome to this non-academic video that how you can do this complicated subject very easily. So I'll be guiding you that you can do this subject conceptually. Don't think that this is a subject where there is compulsion cramming. No, it's not like that. So let's start with this video. My first and foremost point will be that you should not cram and you should go with the concepts and the basics behind it. For example, we say that glycogen is stored mainly in liver for blood glucose maintenance. But why this happens? You should ask a why behind each and everything. Look, we take 2-3 meals in a day, but blood glucose is maintained 24 by 7, right? So in between meals, who's gonna provide blood glucose? It will be easily coming from the liver glycogen. That's why liver glycogen is the form of glucose which is for blood glucose maintenance, right? Then do the things in story form. For example, glycolysis pathway is a sequential pathway of 10 steps, and you can do the pathway in a story form. Like phase 1 is energy utilizing phase. It's like a business, it's like a plan of business where phase 1 is investment of the business. But phase 2 also has 5 steps, and phase 2 is energy producing phase. So there is a why to each and every step, and it becomes so easy. Then I always suggest that don't do unnecessary mnemonics. Only if something is very difficult and there's so many things, only then you make a mnemonics taking the initials of the steps. But otherwise you should do the conceptual things, because if you do more mnemonics, then doing the exam you don't even remember what was the mnemonic, right? Leave the subject. Okay. And mnemonics never help you understand the concepts. That just gives you superficial learning process which you just vomit out during the exam. So I always tell you, minimize your mnemonics. Then you should do formulas in biochemistry. Yes, you'll be surprised on this point, as usually formulas are there in maths and not our medical subjects, and that too biochemistry. But yes, I have made some formulas. I have given them my name, that is SMILE formulas 1 to 4. These formulas help you in understanding the main two hormones work, that is insulin and glucagon. Like questions are asked, which enzyme or pathway is activated by insulin and which is activated or inhibited by glucagon. You can solve all such questions by the formulas which I provide you. Then which enzyme is active in phosphorylated state, which is in dephosphorylated state, that also can be done by the formula. Then fed and fasting state, you should be able to understand the concept of fed and fasting state, and then you can solve all the biochemistry questions which are asked on diabetes, because diabetic situation is same like fasting situation, right? So you apply all the understanding of your fasting state on a diabetic patient and you can solve these questions. Then cell compartment understanding, you have to usually cram that which pathway occurs in cytoplasm, which pathway occurs in mitochondria, right? There's no understanding, but you can understand this concept also, that which pathway occurs in cytoplasm and why. Ask a why behind it and you'll be easily able to learn it. Then there are few small things which makes infrastructure of biochemistry, that you, if you do these things, then biochemistry subject becomes very easy for you. For example, fuel used. What is the fuel used in, for which organ, in which state, fed state or fasting state or starvation state? That is one table given in Harper. You can just do that, right? And it will be very easy for you to solve questions. Then do the enzyme concepts before doing pathways. That is, like enzyme commission numbers are asked, and you should be very clear with what these 6 category of enzymes they are doing, right? Like the first one, enzyme category number 1 is oxidoreductase, second is transferase. You usually learn that, no? So do that, do those concepts before pathways. Then diet concepts help you in understanding fed and fasting state, that how much carbohydrates which we take, that is how much is converted to fats in body and how much is used for energy. Then always do NAD and NADPH very clearly in the starting only, before starting the subject, because usually students get confused that which form of NAD is for energy and which form is for anabolic pathways, right? So you have to do it very clearly. And all these basic concepts required before starting the subject are covered in my book in the starting 9 pages of the book. So this helps you a lot. Then always in biochemistry, interlink the pathways. Few of the pathways are very much interlinked. For example, urea cycle and TCA cycle link occurs with the help of fumarate and aspartate. So always do the links. It helps you do both the pathways together, right? And then glycogen synthesis and breakdown, as they both occur in cytoplasm. Then glycolysis and gluconeogenesis regulation is very much interlinked, as they are opposite pathways. Glycolysis is breakdown of glucose and gluconeogenesis is making of glucose. Then ketone body pathways, ketone body synthesis and ketone body breakdown both occur in mitochondria. Then beta oxidation of fatty acids and fatty acid synthesis. You have to do these pathways very clearly, because usually students get confused between beta oxidation, the steps of beta oxidation and fatty acid synthesis, right? Then energetics in biochemistry. Pathways have energetics. You have to do all the energetics together at one place, right? So that it is very easy for you to revise. Then learn all the regulations together. The insulin glucagon relation and the allosteric and covalent modifications, all these regulations, you should make your notes together in short, so that it is very easy for you to revise. Then you can write all the rate limiting enzymes together on one page and keep revising. All these things are covered separately in my rapid revision notes. Then I will suggest you that you take all the cycles notes on 4-5 pages in your notebook or on simple pages and keep revising it. If you do revise in the morning time, then it will be most beneficial, as told by our elders and many scriptures. Glycolysis, also known as Embden-Meyerhof pathway by the name of scientists who have given it this, also known as EMP pathway, short form EMP pathway. So, you know, HMP is hexose monophosphate pathway, which produces NADPH. That is a minor source, that is a minor pathway of oxidation of glucose. But glycolysis, EMP pathway, is the major pathway of glucose oxidation. So even if EMP written in the option, so you should be knowing that this is glycolysis written, right? So let's start. The first step is glucose gets converted to glucose 6-phosphate. I told you the first step which occurs is always phosphorylation, and this is because of this is for the entrapment of glucose inside the cells, right? So this first step is phosphorylation, glucose to glucose 6-phosphate. Here, for adding this high energy phosphate bond, ATP is used. ATP is used, gets converted to ADP. The enzyme must be some kinase. I told you, whenever something with ADP, ATP, mostly enzyme is a kinase, right? So here ATP used, enzyme is kinase. Here it is hexokinase or glucokinase. The difference between hexokinase and glucokinase we'll be later doing in enzymes topic, right? So the first step is done, hexokinase, ATP is used. Why is glycolysis occurring in cells? The reason is to obtain ATP, but at the very first step we are using ATP. We are doing opposite of the target. Like when opposite of the target is done, this is known as rule of business, because see, in business we want money, right? But at the very first step we have to invest money. So only first when we invest, later we will be able to gain. So glycolysis is also like a business, that first we are adding ATP and after that we will be obtaining ATP. And any good business is that where we invest less and we obtain more, right? So I'll be taking glycolysis as a rule, as a plan of business. The nature has made this plan of business. So let's see how. The second step is glucose to fructose, glucose 6-phosphate to fructose 6-phosphate. Because glucose and fructose are isomers of each other, so here the name of enzyme is isomerase, simply, right? And in the next step, fructose 6-phosphate gets converted to fructose 1,6-bisphosphate. So here we are adding one more phosphate at position number 1 now, making fructose 1,6-bisphosphate. Phosphate is a high energy bond. To add phosphate, we always use ATP. So here also ATP used gets converted to ADP. Enzyme must be some kinase, and that is phosphofructokinase 1. Short form, this third number step, short form PFK1, phosphofructokinase 1. This is the rate limiting enzyme of glycolysis, also the first committed step of glycolysis. What do you mean by committed step? Committed means when this step will occur, then it is very sure that glycolysis will occur. That means first and second steps of this pathway were not committed for glycolysis because glucose 6-phosphate and fructose 6-phosphate, they can be used for other pathways also. But fructose 1,6-bisphosphate, when this compound is formed, fructose 1,6-bisphosphate, then we are very sure that now glycolysis will occur, pyruvate will be formed, right? So therefore I say PFK1 is the committed step, first committed step of glycolysis, right? Now let's see the fourth number step. Fourth number step is done by enzyme aldolase A. Write down A here, aldolase A. Where is aldolase B? Aldolase B will be doing later in fructose metabolism, right? Aldolase A is in glycolysis. The substrate for aldolase A is fructose 1,6-bisphosphate, whereas the substrate for aldolase B is fructose 1-phosphate, right? Let's see what aldolase A is doing. Aldolase A will break down this compound. This is a 6-carbon compound with 2 phosphates, which is now broken down to 2 3-carbon compounds. One is aldehyde sugar, other is keto sugar. Both are given 1,1-phosphate each. So let's see. Fructose 1,6-bisphosphate gets converted to dihydroxyacetone phosphate. This is 3-carbon keto with a phosphate, dihydroxyacetone phosphate, the short form DHAP. Later I'll be using the short forms. DHAP is formed. On the other hand, 3-carbon aldehyde is glyceraldehyde 3-phosphate. So this 3-carbon aldehyde is also given 1 phosphate. So now we have got DHAP and glyceraldehyde 3-phosphate as the end products of number 4 step. Now number 5th step is DHAP also gets converted to glyceraldehyde 3-phosphate. This way we get 2 molecules of glyceraldehyde 3-phosphate, and the name of 5th number enzyme is this aldehyde and keto interconversion is always by isomerase. So number 5th enzyme is isomerase. Now see this glycolysis pathway as a whole. There are 10 steps in glycolysis. These first 5 steps we are done. These first 5 steps constitute phase 1 of glycolysis, also known as energy utilizing phase of glycolysis, or you can say investment of business. This is the investment of business, right? And phase 2, the next 10 steps come in phase 2, and that is energy producing phase of glycolysis. Energy producing phase, right? Now see one smartness planned by nature. I told you that any good business is that we invest less and we obtain more, right? Now see phase 1. In phase 1, how many molecules are entering? How many glucose are entering phase 1? Only 1 molecule of glucose is entering. And therefore, whenever we use ATP in phase 1, we have to use it for 1 molecule. But in phase 2, how many molecules are entering phase 2? 2 molecules of glyceraldehyde 3-phosphate are entering. So that in phase 2, when we obtain energy, then every energetics is multiplied by 2 because 2 molecules are entering. This is a smartness of business planned here, that investment is less but we obtain more energy. Now let's see the detail of phase 2. The first step in phase 2 is number 6 step, that is glyceraldehyde 3-phosphate gets converted to 1,3-bisphosphoglycerate. This is the master plan of glycolysis. This step is very special step in glycolysis. Understand this very carefully. See here, glyceraldehyde 3-phosphate is getting converted to 1,3-bisphosphoglycerate. Can you see in this step one more phosphate is coming at position number 1? this phosphate, we are adding phosphate, high energy bond. Usually to add phosphate we use ATP, but in this step we are not using ATP because we are in energy producing phase, we are in phase 2. So we will not use ATP here. So this phosphate is simply taken from inorganic phosphate. Inorganic phosphate is always there in the medium, so we can take it. But generally, I repeat, generally whenever we add phosphate we use ATP, but this step is very, very special step where ATP is not used but phosphate is added. This is a very, very special and rare step which can add phosphate without using ATP, right? And the name of this enzyme is glyceraldehyde 3-phosphate dehydrogenase, and this is the only step of glycolysis which can use inorganic phosphate. And see, the name of enzyme is dehydrogenase. I told you whenever dehydrogenase means hydrogen removed and somebody will come to take this hydrogen, that somebody most of the time is NAD. Here also it is NAD. NAD will take this hydrogen, forms NADH. I have written 2 here because we started with 2 molecules of glyceraldehyde 3-phosphate. These 2 NADH will later go into ETC to give 5 ATPs. So this step is responsible for giving 5 ATPs. See, this is such a special step. In this step, we are adding phosphate, and to add phosphate, we generally use ATP, but this step is not using ATP. In fact, it is producing ATP, and that too 5 ATPs. Till now, we have just used 2 ATPs and we have obtained 5. It is such a smart plan of business, and many more ATPs are yet to come. Now let's go further. We have reached till this point 1,3-bisphosphoglycerate. Now this 1,3-bisphosphoglycerate is a 3-carbon molecule having 2 phosphates. Now only one work is left in glycolysis, that is, we have 2 phosphates. Phosphate is a high energy bond. Now we will keep breaking these 2 phosphates one by one, and when we break high energy bond, then energy is released. That energy can be trapped in the form of ATP, right? Only this work is left. So let's see. 1,3-bisphosphoglycerate gets converted to 3-phosphoglycerate. So you can see we are removing phosphate from number 1 position. 1,3 gets converted to 3. We are removing phosphate from number 1 position. Who will take this phosphate? ADP will take this phosphate, gets converted to ATP. I have written 2 here because we started with 2 molecules. And whenever ATP formed at some step, immediately there and then only ATP formation occurs, known as substrate level phosphorylation. And enzyme of substrate level phosphorylation is always a kinase. And here it is phosphoglycerate kinase. Short form is PG kinase. PG kinase is a substrate level phosphorylation step of glycolysis. They ask you many questions on substrate level phosphorylation steps. So it is very easy to learn these steps. You have to learn the substrate, you have to learn the product, you have to learn the enzyme of such a step, right? Now we have reached at this point 3-phosphoglycerate. Now we have our last phosphate attached at number 3 position. We can remove this phosphate also and we can obtain energy. But there is a problem. When 3 number phosphate is attached at glycerate, this is not a high energy bond. This is not a very high energy bond. If I break this bond here, then I won't be able to get enough energy to make ATP, right? So what we have to do is that first we have to make this bond high energy and then we will break this last high energy bond. So our next 2 steps, that is number 8th and number 9th step, is like a preparation to make high energy bond. So let's see the detail. 3-phosphoglycerate gets converted to 2-phosphoglycerate. So phosphate is getting transferred from number 3 position to number 2 position. And the name of enzyme is mutase. So what is the EC number of mutase? Transfer of phosphate is occurring from position number 3 to 2, but mutase is not EC number 2 transferase. No, it is not transferase, because in this step molecular formula is not changed. One isomer is getting converted to another isomer. Transfer is occurring, but within the molecule, intramolecular transfer is occurring, and whenever in cells intramolecular transfer is occurring, be it methyl group, sulfate group, phosphate group, the enzyme used is always mutase, and mutase belongs to isomerase category, that is EC number 5, not EC number 2. Be careful with this. Now next step, 2-phosphoglycerate gets converted to phosphoenol pyruvate. Enol. Focus here. En means double bond, ol means OH.
So this kind of bond with double bond and OH is made here. So you can just take an idea that this is a high energy bond. And to make this enol high energy bond, which enzyme is used? Enolase, right? So to make enol bond, it is enolase enzyme, very easy, right? Now, phosphoenol pyruvate we have obtained, that is, we have our last phosphate attached to pyruvate with high energy enol bond. We can break this enol bond and we can get pyruvate. On the other hand, phosphate is taken by ADP, gets converted to ATP. I have written 2 here because we started with 2 molecules. Whenever ATP formed at such a step known as substrate level phosphorylation step, the enzyme must be a kinase, and here it is pyruvate kinase. Here it is pyruvate kinase. Now pyruvate is the end product of aerobic glycolysis, but lactate is formed in case of anaerobic glycolysis, and here the enzyme is lactate dehydrogenase, short form LDH I have written, lactate dehydrogenase. Can you tell me where is pyruvate dehydrogenase? Pyruvate dehydrogenase is in link reaction, right? But this lactate dehydrogenase is in anaerobic glycolysis. So we are done with the pathway and all the 10 to 11 enzymes we are done. Now I'm going to write the EC numbers of all the enzymes. See, hexokinase EC number is 2 because it is a kinase. Isomerase EC number is 5. Then PFK1, it is a kinase, so EC number 2. Aldolase A, it is breaking the bond without using water. That's why EC number is 4, right? 4 is lyase, bond is made or broken, water and ATP not used. So aldolase A or aldolase B both are in EC number 4 category. Then 5th number enzyme is isomerase, so EC number is also 5. Then 6th number enzyme is dehydrogenase. Dehydrogenase means EC number 1. Then PG kinase, so 2. Mutase, 5 I told you. Enolase is EC number 4. This I will explain later in enzymes topic. Then pyruvate kinase, because it is a kinase, so EC number 2. And the last one, lactate dehydrogenase of anaerobic glycolysis, EC number is 1 here, right? Now there are 3 irreversible steps in glycolysis. These are, you can see here, broad black arrows I have made. These broad black irreversible arrows I have made. So these 3 enzymes, these 3 steps are irreversible or regulatory steps of glycolysis, that is hexokinase, PFK1 and pyruvate kinase. So you can note here that all these 3 are kinases, right? Hexokinase, pyruvate kinase, phosphofructokinase. So these 3 regulatory steps are kinases. Now there are total 4 kinases in glycolysis. One extra is PG kinase. So can I say that all kinases of glycolysis except phosphoglycerate kinase are irreversible or regulatory steps of glycolysis? And now let's see the substrate level phosphorylation enzymes of glycolysis. That is this and this, that is PG kinase and pyruvate kinase. They are substrate level steps of glycolysis. Now you can see pyruvate kinase enzyme is coming in the list of irreversible regulatory enzymes also, and pyruvate kinase is also substrate level phosphorylation enzyme. This enzyme is common in both the list. I told you something special about pyruvate kinase enzyme. You have done that. Pyruvate kinase enzyme requires potassium more than magnesium. Most of the kinases they use magnesium, but pyruvate kinase requires potassium more than magnesium. Now one more point about pyruvate kinase is that this enzyme deficiency is the second most common human enzyme deficiency. There is one more enzyme which is the first most common human enzyme deficiency, that is G6PD enzyme, which is enzyme of HMP pathway. Pyruvate kinase enzyme is of EMP pathway or glycolysis. G6PD is enzyme of HMP pathway, and this is the first most common human enzyme deficiency. Now both these patients, that is pyruvate kinase deficient and G6PD deficient patients, they both are very common. See, first most common and second most common enzyme deficiency. They are common and they have one common clinical feature, that they both present with hemolysis. So how will you distinguish between these two patients? There's a simple thing, Heinz bodies. You know, Heinz bodies are present in G6PD deficiency. So if hemolysis and Heinz bodies are present, then it is a G6PD deficient patient. If hemolysis is present but absent Heinz bodies, then most probably it is pyruvate kinase deficiency.
Now let's see how hemolysis occurs in deficiency of pyruvate kinase also, and why hemolysis occurs in G6PD deficiency. In G6PD deficiency, hemolysis occurs due to oxidative stress, because normal role of G6PD in RBCs is to make NADPH, which will make reduced glutathione, which will destroy H2O2. But if G6PD is not there, then H2O2 is not destroyed and it will start accumulating in RBCs, leading to damage to the membrane of RBCs, leading to hemolysis, right? And why pyruvate kinase deficiency leads to hemolysis? Because pyruvate kinase or any enzyme of glycolysis, if deficient, then glucose is unable to give energy. Glucose is unable to give energy, and in our body, RBCs are such cells which depends on glucose, whether body is in fed state or fasting state or starvation state. They are only dependent on glucose. RBCs cannot use any other fuel for energy. So when glucose is unable to give energy, then there are no ATPs in RBCs. So sodium potassium ATPase pump will be affected, leading to hemolysis. So this is the reason of hemolysis in G6PD and pyruvate kinase deficiency. The reason is different in both the conditions, right?
Now let's do the energetics of glycolysis. One glucose molecule gets converted to two molecule of pyruvate. Pyruvate is a 3-carbon compound. So from 6-carbon glucose, we get 2 molecules of 3-carbon pyruvate. And here from glucose to pyruvate conversion, there are 10 steps. In these 10 steps, how many ATPs are used? Minus 2 ATPs, that is 2 ATPs are used. 1 ATP used at hexokinase step, other ATP used at phosphofructokinase 1 step. And there are 2 steps of substrate level phosphorylation which are producing 4 ATPs. So I'm writing plus 4 ATPs. So ATP thing I have written on left hand side. On right hand side I will write about NAD, NADH. And you know, in glycolysis there is one step, glyceraldehyde 3-phosphate dehydrogenase, where NADH is produced. So NAD gets converted to NADH, and here this occurs in phase 2. So I have written 2 here. 2 NAD forms 2 NADH. These 2 NADH will later go into ETC to give 5 ATPs. So the full energetics is 5 plus 4, that is 9, minus 2, that is 7 ATPs we get in case of aerobic glycolysis, right? But in case of anaerobic glycolysis, these 2 pyruvate gets converted to 2 lactate, and this is 11th number step of glycolysis we can say, and the name of enzyme is lactate dehydrogenase. You know, whenever dehydrogenase, then somebody will come to take hydrogen, mostly NAD forms NADH. But lactate dehydrogenase is an opposite dehydrogenase. Here NADH will form NAD, right? So opposite will occur here. That is, in this step, this NADH which was formed earlier, that will be converted to NAD. So now the energetics of anaerobic glycolysis will be, these 5 ATPs are not obtained because this NADH has formed NAD. So we can't use this NADH for ETC. So these 5 ATPs are not obtained. So energetics will be only this 4 minus 2, that is 2 ATPs. So we get 2 ATPs in case of anaerobic glycolysis, and we get 7 ATPs in case of aerobic glycolysis. Now one very important question is asked. You can note here that there are 10 steps in aerobic glycolysis, but there are 11 steps in case of anaerobic glycolysis. So they can ask you that what is the purpose of this extra step of anaerobic glycolysis? Is the purpose formation of lactate? No, lactate is just the side product. It is just the dead end of glycolysis. That is not the purpose of this step. In fact, the purpose of this step is formation of this NAD. Why? See, if NAD is not formed, then what will happen? Whenever glucose forms pyruvate, then at the time NAD forms NADH. Glucose forms pyruvate, NAD forms NADH. So after some reactions, NAD of the cell will be finished. So now glucose cannot be converted to pyruvate. So we need NAD actually. So this extra step of anaerobic glycolysis is for the regeneration of this NAD so that it can be used again for conversion of glucose to pyruvate. So this step is for regeneration of NAD. Now if I ask you how many ATPs are formed in RBC in aerobic state, what is the answer? Answer is 2 ATP. See how? RBC have no mitochondria. When mitochondria is not there, that NADH which give 5 ATPs via ETC occurs in mitochondria. So this NADH can never give those 5 ATPs in case of RBC. So in RBC, energetics is always 4 minus 2, that is 2 ATPs. In the question, even if they write fed state, fasting state, aerobic state, anaerobic state, they're just using these words to confuse you. But whenever question on RBC, you will say 2 ATPs, right? Now, if they ask you which molecule is dead end of glycolysis, I told you dead end of glycolysis is lactate. What that means? Dead end means once lactate is formed, after that nothing can be formed. Let's see the example. Let's say this is muscle and let's say this muscle is exercising muscle. This muscle is exercising muscle and let's say this is liver here. In the center, this is blood, right? Now, during exercise, anaerobic conditions are created so that in this muscle cell, glucose will form pyruvate and then pyruvate will form lactate because of anaerobic glycolysis. Now, lactate is the dead end of glycolysis, means when lactate is formed, after that nothing can be formed. That is, in this muscle cell where lactate is formed, in this cell, lactate is the dead end. But in body, it is not the dead end. That means in body, it can be converted to some other thing. That is, see, now lactate has to come out of this muscle cell into the blood. From blood, it is taken up by liver. In liver, lactate will form glucose. By which pathway? Lactate forms glucose by gluconeogenesis. And this glucose is again supplied to that exercising muscle. So this is a cycle formed known as Cory's cycle, also known as glucose lactate cycle. Now there is one more similar cycle similar to Cory's cycle, and that is glucose alanine cycle. Let's see what happens there. In case of glucose alanine cycle, let's say this is muscle and now this muscle is fasting muscle. And let's say this is liver, again in the center this is blood. So what happens here, in fasting muscle, glucose will form pyruvate and then pyruvate will form alanine. This alanine comes out in blood. From blood it is taken up by liver. Alanine in liver again gets converted to glucose by pathway gluconeogenesis, and this glucose is again supplied to the fasting muscle. This is known as glucose alanine cycle and also known as Cahill cycle. Cahill, Cahill cycle. So can I say that Cory's cycle occurs in exercising muscle and Cahill cycle occur in fasting muscles, right? Now see one thing here, in liver, alanine is forming glucose by gluconeogenesis. This is one amino acid which is forming glucose. Actually, many amino acids, that is 18 amino acids can form glucose, But you can see here alanine forming glucose. Alanine is most glucogenic amino acid. It is a 3-carbon amino acid, and mostly 3-carbon compounds are good substrates for gluconeogenesis. You can see lactate is also 3-carbon, pyruvate is also 3-carbon, which is forming glucose here by gluconeogenesis. Mostly 3-carbon compounds are good substrates, and alanine amino acid is the most glucogenic amino acid out of 18 amino acids which can form glucose. So now we are going to do RL shunt, that is Rappaport-Lubering shunt, which occurs only in RBCs. Let's see what happens here. First, see normal glycolysis. You know, in normal glycolysis, there is glucose gets converted to pyruvate, and there is one substrate level phosphorylation step occurring here, that is 1,3-bisphosphoglycerate gets converted to 3-phosphoglycerate. Right? This step occurs with enzyme phosphoglycerate kinase. It is a substrate level phosphorylation step which will make 2 ATPs at this step, right? And after 3-phosphoglycerate, there are many steps, and then in the end, pyruvate is formed. Now, what is RL shunt? In RL shunt, this substrate level phosphorylation step is not occurring. Glycolysis is occurring, just that this step is not occurring. Instead of this step, there is a shunt which is followed. So, instead of this straight route, this shunt route is followed. Let's see how. 1,3-bisphosphoglycerate gets converted to 2,3-bisphosphoglycerate, and then 2,3-bisphosphoglycerate forms 3-phosphoglycerate, and after that, after next steps are same and pyruvate is formed. So this shunt is followed, and that is known as RL shunt. Let's see the enzymes here. When 1,3 is converted to 2,3-bisphosphoglycerate, there is intramolecular transfer or shifting occurring. This is enzyme is mutase. When 2,3-bisphosphoglycerate gets converted to 3-phosphoglycerate, there is removal of phosphate. You know, the enzyme for removal is phosphatase, right? So, RL shunt, that is Rappaport-Lubering shunt or cycle. Rappaport-Lubering shunt or cycle. This occurs only in RBCs. When this occurs only in RBCs, so how many ATPs will be obtained in RL shunt? You know, in RBC, ATPs obtained are 2. Be it any case, ATPs obtained are 2. But in RL shunt, we are losing 2 ATPs because of one step substrate level phosphorylation step of PG kinase not occurring in this case. So 2 minus 2 is 0. So number of ATPs obtained, net gain of ATPs in RL shunt is 0. There is one question in your guide. They ask you that how many ATPs are formed in RL shunt? 0 option is not there, right? So now you can mark 2. Listen to the question again. How many ATPs are formed in RL shunt? So you can say 2 are formed. But when 0 option is there, then best is to mark 0 option. But if 0 option is not there, then you can mark 2, right? See, whenever questions on energetics are asked, then by default, you have to consider net gain of ATP they are asking, right? But if this net gain of ATP, this thing answer is not given, then you can consider the other way around, right? So I can say here that full story of RBC is this. The full story of RBC is this, that whatever glucose is entering into RBCs, 90% of glucose out of this is going into normal glycolysis to give you 2 ATPs per glucose molecule, right? But 10% of this glucose, 10% of glucose entering into RBC is not going into normal glycolysis. You can say it is going into RL shunt, so that this 10% glucose is not forming any ATP, because this 10% glucose purpose is not to make ATP. Instead, its purpose is formation of 2,3-BPG, right? So, can I say that number of ATPs formed in RBC can never be more than 2, but it can be less than 2 if it is RL shunt, right? So what is the use of this compound 2,3-BPG in RBCs? This 2,3-BPG is used for the release of oxygen from hemoglobin A. It can only affect hemoglobin A because hemoglobin A is alpha to beta 2, 2 alpha to beta chains, And this compound 2,3-BPG binds to beta chain and affect the release of oxygen. So therefore, it will not affect any other hemoglobin. Any other hemoglobin like fetal hemoglobin HbF is alpha 2 gamma 2. There is no beta chain in any other hemoglobin and 2,3-BPG binds to beta chain. Therefore, 2,3-BPG will not affect any other hemoglobin, right? And also, fetal hemoglobin should not be affected because fetal hemoglobin should take oxygen from mother's blood. It should not release oxygen, right? Now tell me, how many irreversible steps are there in glycolysis? In glycolysis, there are total 10 to 11 steps. Out of them, 3 are irreversible, rest 7 or 8 are reversible steps. So tell me, glycolysis pathway is reversible or irreversible? Yes, glycolysis pathway is irreversible. Even if one step is irreversible, then the pathway is irreversible. So glycolysis is irreversible. Next, tell me, is glycolysis complete breakdown of glucose? No, glycolysis is not complete breakdown of glucose. Complete breakdown of glucose is C6H12O6 plus 6 oxygen will give 6 water and 6 carbon dioxide. These water and carbon dioxide will be released few in link reaction TCA, ETC. So when link reaction TCA, ETC also occurs, then it is complete breakdown of glucose when all these 6 carbon dioxide and water are released from glucose, right? So glycolysis is not complete breakdown of glucose. Now let's take the inhibitors of glycolysis. Inhibitors of glycolysis. First is iodoacetate. Iodoacetate will inhibit the enzyme glyceraldehyde 3-phosphate dehydrogenase. Second is arsenate. This also inhibits glyceraldehyde 3-phosphate dehydrogenase. Same enzyme is inhibited. Third is sodium fluoride, which inhibit enolase enzyme. And the sodium fluoride is also used in blood glucose estimation for inhibition of glycolysis, so that when we are measuring glucose in lab, in vitro condition, then the cells should not use glucose by glycolysis, because if cells will keep using glucose in that blood sample, then by the time lab will check the glucose, by that time glucose will be falsely decreased. It will not represent the true glucose value which was there in the patient, right? So therefore, we have to use the inhibitor of glycolysis in the sample when we are taking the sample for blood glucose estimation. The fourth inhibitor I am going to write is oxamate. This is not oxalate, this is oxamate, which is inhibitor of enzyme lactate dehydrogenase. So I have told you these 4 inhibitors of glycolysis. Inhibitor of glycolysis means when these inhibitors are there, then glycolysis pathway will be inhibited, right? Inhibitor means pathway will be inhibited, right? But there is one special thing for arsenate. When arsenate act as inhibitor of glycolysis, then this pathway glycolysis is not inhibited. Glycolysis will continue. The glycolysis pathway will continue. Pyruvate will be formed, but just that no ATP will be formed. That is 0 ATP formed in arsenic poisoning in glycolysis, right? And glycolysis occurs in which compartment of the cell? It occurs in cytoplasm. But after glycolysis, we have link reaction, TCA and ETC. All the other 3 things, link reaction, TCA, ETC, they all occur in mitochondria. So this is it with glycolysis, Krebs cycle or TCA cycle. First, we'll be understanding the names. One name is TCA. Why the name TCA? The full form is tricarboxylic acid. By the name tricarboxylic, tricarboxylic means 3 COOH group, 3 carboxy group present in some compound. And let me tell you, the first 2 compounds formed in TCA, that is citrate and isocitrate, they are tricarboxylic acids, right? Now, the other name is Krebs cycle by the name of the scientist. So, Krebs cycle if written means TCA cycle. If Krebs-Hanslet cycle written, be careful. If Krebs-Hanslet cycle written, that is not TCA cycle, that is urea cycle, right? Now, there's one more name of TCA cycle, that is citric acid cycle, because the first compound formed in citric acid cycle is citrate. That's why the name citric acid cycle, because the first compound formed is citrate. Now, what is the difference between citric acid and citrate? Citric acid is COOH, acid group, COH group. Citrate means the ionized form, that is COO minus, right? In body, everything is ionized. So, in body, it is present as citrate, right? Now, TCA cycle, tell me, it is anabolic or catabolic pathway? It is both anabolic as well as catabolic, and therefore it is known as amphibolic pathway. TCA is amphibolic pathway. Like both polar, non-polar known as amphipathic, similarly, both anabolic, catabolic is known as amphibolic. When I say TCA is catabolic, what that means? What is the catabolic role of TCA? Catabolic role of TCA means all the macromolecules, carbohydrates, fats and proteins, they all, when they are getting broken down in body, they all form acetyl-CoA, which enters TCA cycle. So, can we say that all macromolecules are getting broken down and they finally reach TCA cycle? So, TCA cycle is the common catabolic pathway, right, of all the macromolecules, because all the macromolecules gets broken down and reach TCA cycle. Now, what is anabolic role of TCA? Anabolic role of TCA, anabolic means synthesis, because many TCA intermediates are used for the synthesis of important compounds in the body. TCA intermediates are used for synthesis of many compounds in the body. For example, succinyl-CoA is intermediate of TCA cycle, which is used for the formation of heme in the body. If you remember, the first step of heme synthesis is succinyl-CoA plus glycine makes ALA, that is delta aminolevulinic acid, right? So, intermediate of TCA cycle is used for the synthesis of some important compound in the body. So, therefore, this is anabolic role of TCA cycle, right? Also, TCA cycle is a vital cycle for the cells. Vital cycle, because it is a vital cycle, it always has to occur. Therefore, there is no enzyme deficiency of TCA cycle. This kind of topic is not there in your books. Enzyme deficiency of TCA, not there, because it is a vital cycle. It always has to occur. Like there's no life without TCA cycle, right? See, on one hand, I'm saying TCA cycle is vital cycle. That means all the TCA intermediates should always be present in any cell of the body. They should be maintained at constant level. And on the other hand, I'm saying that anabolic role of TCA, those reactions are using TCA intermediates. So, that means there must be some reactions which are producing TCA intermediates, because anabolic role of TCA is using TCA intermediates. So, there are some reactions which produce or which replenish TCA intermediates. Those are known as anaplerotic reactions. So, anaplerotic reactions are those reactions which replenish TCA intermediates, For example, pyruvate can form oxaloacetate. OAA when I write, that is the short form of oxaloacetate. See how this reaction will occur. Pyruvate is a 3-carbon compound, oxaloacetate is a 4-carbon compound. So from 3 to 4 carbon, we can simply add carbon dioxide, and the name of this enzyme is pyruvate carboxylase. So pyruvate carboxylase, which is synthesizing oxaloacetate, this is the most important anaplerotic reaction. Now, solve this question. TCA is activated by which hormone? Insulin, glucagon, both or none? I told you TCA is amphibolic, both catabolic and anabolic. You know anabolic pathways are activated by insulin, catabolic pathways are activated by glucagon. So, most of the students will answer both for this question. But answer is not both, answer is none. Because TCA is not having any hormonal control because it is a vital cycle of the body. It does not depend upon hormones. See, when I say TCA is activated by insulin, it would mean that when we are in fed state, when we are eating food, then TCA will occur. And if I say it is activated by glucagon, then it would mean that TCA is occurring when we are in fasting state. But no, TCA does not depend on our food status. It is a vital cycle for the cells. It will always occur, does not depend on food status. So I will say that TCA has no hormonal control because it is a vital cycle for the cells. Now, when I say it is not controlled by hormones, so that doesn't mean TCA will just go on and on and on till infinity. No, there's some control, right? Something has to control TCA cycle. So let's see, TCA is controlled by which thing? TCA is controlled by, see, mainly TCA in the cells is for energy. So directly energy status is controlling TCA cycle. So energy status of the cell. That is, if a cell has not any ATP, the cell is not having ATP, then TCA will occur, right? But if a cell has enough ATP, then TCA will not occur, right? So simply it directly depends on energy status of the cell. And second, it depends on availability of oxaloacetate. Sometimes students say it depends on availability of acetyl-CoA, right? But no, it is not acetyl-CoA. It depends on the availability of oxaloacetate. That's why oxaloacetate is regarded as carrier of TCA cycle. Oxaloacetate is regarded as first substrate of TCA cycle. And also oxaloacetate has catalytic role in TCA cycle. When I say catalytic role, what that means? See, enzymes have catalytic role. Enzymes are catalyst. What that means? That enzyme is not used up in the reaction, right? Enzyme is obtained as such after the reaction. Similarly, oxaloacetate is again regenerated in TCA cycle. It is not used up in TCA cycle. See what that means? Catalytic role. Oxaloacetate will combine with acetyl-CoA as the first step of TCA cycle, making citrate. After citrate, there are so many steps. Finally, oxaloacetate is again regenerated back. So we say that this oxaloacetate is having catalytic role in TCA cycle. Because oxaloacetate has a catalytic role, therefore TCA is not a pathway. It is a cycle. See, because oxaloacetate is again obtained. So it is not a pathway. Pathway means going in one direction like glycolysis, right? Glycolysis is a pathway, glucose forms pyruvate. But in TCA, we are starting from oxaloacetate as the first substrate. In the end also we are getting oxaloacetate. So we say that TCA is a cycle. It is not a pathway. Right? Now let's see few compounds. For example, glutarate. Tell me what is the difference between glutarate and glutamate. Glutarate, we do know alpha ketoglutarate intermediate of TCA cycle. Glutamate is amino acid. There is one more, glutamine. So the names are matching here. So you have to be very clear for the difference in them. The similarity is that all these three things have 5 carbons. Now it is very easy to learn the difference when I have written these 3 compounds like this. On right hand side, make arrow and keep on adding amino group. So if you add one amino group in alpha ketoglutarate, it becomes glutamate. You add one more amino group, it becomes glutamine, right? Similarly, oxaloacetate will form aspartate, will further form asparagine. Now all these compounds, they have 4 carbons, and again same, keep on adding amino group, that is add one amino group in oxaloacetate, it will become aspartate. Add one more amino group in aspartate, it will become asparagine. Now see here, can you see these 4 are amino acids? Glutamate, glutamine, aspartate, asparagine. These 4 amino acids are getting synthesized from TCA cycle intermediates. I told you this is anabolic role of TCA, that TCA intermediates are used for the synthesis of important things in the body, And here you can see 4 amino acids, obviously non-essential amino acids, they are getting formed in body from TCA intermediates. So this is anabolic role of TCA cycle. Also, you have to note that succinate or succinyl-CoA, this compound is also having 4 carbons, right? Then let's see the difference between malonate. There is a compound malonate, other is malate. Malonate, when this N is there, this is a 3 carbon compound, and malate is a 4 carbon compound. Malonate is inhibitor of TCA cycle and malate is intermediate of TCA cycle, right? Now this compound malonate or malonyl-CoA, this compound, this is a 3 carbon compound, you know, it is inhibitor of 3 pathways in the body. One is TCA cycle, ETC and beta oxidation of fatty acid. Actually, it inhibits succinate dehydrogenase of TCA cycle. It will inhibit complex 2 of ETC and it inhibits CPT1 of beta oxidation of fatty acid. What is the full form of CPT? It is carnitine palmitoyl transferase 1, right? And also tell me, what is the full form of CPS enzyme? CPS is carbamoyl phosphate synthetase 1 and 2 we have. CPS 1 is in urea cycle, CPS 2 is in pyrimidine synthesis, right? But this I have written is CPT carnitine palmitoyl transferase 1 and 2, which are there in beta oxidation of fatty acid. Now malonate inhibits CPT1 of beta oxidation of fatty acid, right? Then note down, all the enzymes of TCA cycle are lying in mitochondrial matrix except one, and that is succinate dehydrogenase, which is lying in inner mitochondrial membrane. You know, in inner mitochondrial membrane, ETC occurs. This enzyme has also a role in ETC, that is complex 2 of ETC, that we'll be seeing later in another video for ETC, right? Now let's start with the cycle, Krebs cycle. You know the starting material or the first substrate we say, first substrate is oxaloacetate. I told you oxaloacetate is 4 carbon compound. This oxaloacetate will combine with acetyl-CoA, which is 2 carbon. They both join to make citrate. So 4 plus 2, this is 6 carbon compound. And the name of enzyme is citrate synthase. I told you citrate synthase belongs to transferase category, that is EC number 2, right? Next is citrate gets converted to its isomer, that is isocitrate. So this is also 6 carbon compound. And name of enzyme is aconitase. This aconitase is converting one isomer to another. So EC number should be 5 here, but it is not 5. Aconitase is a lyase, that is EC number 4. Also note down fumarase is also a lyase, right? Now see the next 2 steps of TCA cycle. This step and this step, that is isocitrate to alpha ketoglutarate to succinyl-CoA. These 2 steps are same like we have done in link reaction. You remember link reaction was 3 carbon to 2 carbon, removal of carbon dioxide, name of enzyme is dehydrogenase. So that was oxidative decarboxylation requiring B1 and name of enzyme is dehydrogenase here, and also NAD forms NADH, right? Next 2 steps of TCA are same like link reaction, that is 6 carbon isocitrate gets converted to 5 carbon alpha ketoglutarate, then this 5 carbon gets converted to 4 carbon succinyl-CoA. So in both the steps, carbon dioxide is getting removed. Name of enzyme is dehydrogenase. The first one is isocitrate dehydrogenase. The next one is alpha ketoglutarate dehydrogenase. In both these steps, B1 vitamin used, oxidative decarboxylation, and NAD forms NADH, right? So we have reached till this point, succinyl-CoA. Now see here, we are done with this half of TCA. In this half of TCA, we have done 2 dehydrogenases. They are doing oxidative decarboxylation. And see this, now this half of TCA, where we are starting from succinyl-CoA, and after all these reactions, we are ending at oxaloacetate. Oxaloacetate is also 4 carbon, succinyl-CoA is also 4 carbon. So all the intermediates in between these 2 are 4 carbon compounds. Succinate, fumarate, malate, all are 4 carbon compounds. And whenever dehydrogenase on this side of the cycle, see succinate dehydrogenase, malate dehydrogenase, there is no change in number of carbons when these two dehydrogenases are working. So they both are simply oxidation steps occurring. You know, dehydrogenase means removal of hydrogen, means oxidation, right? Now let's see the next step in detail, that is succinyl-CoA gets converted to succinate. You know that CoA is a high energy bond. Now this bond is getting broken in this step. Succinyl-CoA gets converted to succinate. So CoA is a high energy bond. When this bond is broken, energy is released. That energy can be trapped in the form of ATP or GTP. And here GTP is written, and you know ATP GTP formed at some step is known as substrate level phosphorylation step. In glycolysis, we have done two substrate level phosphorylation steps, but in TCA cycle, this is the only substrate level phosphorylation step occurring. And you know, enzyme must be some kinase. Here it is succinate thiokinase. So what is the EC number here? See, I told you if it is kinase, then EC number is 2. But if thiokinase is written, then EC number is 6. Right? Be careful. If thiokinase is written, then EC number is 6. Right? Now, in this step, whenever you see this diagram TCA cycle in any of the book, then here in this step, they write GTP formed, right? But let me tell you, this is wrong. Actually, most of the time, this enzyme will form ATP and not GTP. Most of the time, it will form ATP. If you will see your latest edition of Harper, 30th edition, in this book, they have done this correction that in the diagram, they have written ATP here because most of the time, this enzyme forms ATP. So, if they simply ask you a question, thiokinase produces what thing? Thiokinase produces what? You will say ATP or GTP? You will say ATP because I told you most of the time this enzyme will form ATP. But if they ask you thiokinase in liver, in kidney, and if they ask you during starvation, if these three conditions they ask you in liver, in kidney, during starvation, it produces what? Then you will say GTP. Understand why? Because during starvation, this pathway occurs, gluconeogenesis occurs. Actually, this pathway gluconeogenesis requires GTP, and gluconeogenesis, you know, occurs in these two organs, liver and kidney. So, can I say that thiokinase of TCA produces GTP in liver and kidney during starvation for gluconeogenesis, right? You know, gluconeogenesis is a high energy consuming pathway. This pathway requires GTP and that is provided by thiokinase of TCA cycle, right? So we have reached till this point, succinate. Let's see the next step. Succinate to fumarate. There is no change in number of carbon. Enzyme is dehydrogenase, that is succinate dehydrogenase. So this is simple oxidation step occurring and somebody will come to take this hydrogen. That somebody here is FAD. FAD forms FADH2, right? So can I say in this step, FAD means vitamin B2 is used. Succinate dehydrogenase will use vitamin B2, right? And you know, if NAD means B3, if oxidative decarboxylation means B1, if simple decarboxylation means B6, right? All that you know. So let's see the next step. Fumarate to malate. Enzyme is fumarase. I already told you it is a lyase. And then malate to oxaloacetate, the last step of TCA cycle. There is no change in number of carbons. Enzyme is malate dehydrogenase. So there's simple oxidation occurring. And here NAD comes to take hydrogen and forms NADH, right? So we are done with the detailed steps of TCA cycle. Now let's again see TCA cycle with the eagle's eye view. That is, see what is happening. But other substances are intermediate of TCA cycle, which I have explained, which I have written in the circular form. Oxaloacetate, citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate and malate. They all are intermediates of TCA cycle, but acetyl-CoA is not the intermediate of TCA cycle. Now let's see the energetics of TCA cycle from 1-acetyl-CoA. If 1-acetyl-CoA enters TCA cycle, then how many NADH obtained? One here at malate dehydrogenase, one at alpha-ketoglutarate dehydrogenase and one at isocitrate dehydrogenase. So 3 NADH, 1 FADH2 and 1 ATP or GTP here. So total how many ATPs? 3 NADH, 3 into 2.5 is 7.5. 1 FADH2 will give 1.5 and 1 ATP or GTP gives 1 ATP. So this is total 10 ATPs per acetyl-CoA. So these 10 ATPs are from one acetyl-CoA. Now let's count from one glucose, and that will be complete breakdown of glucose. Now we are going to count. So one glucose will be converted to 2-pyruvate. 2-pyruvate via link reaction will make 2 acetyl-CoA, and these 2 acetyl-CoA will go into TCA cycle, right? So energetics is in glycolysis, glucose to 2-pyruvate glycolysis, we get 7 ATPs because it is aerobic glycolysis, end product pyruvate. Then in link reaction, I told you 2 NADH are formed. So 2 NADH will give 5 ATPs, and when 2 acetyl-CoA will go into TCA cycle, then they will give 20 ATPs. So this total is 32 ATPs from complete breakdown or complete oxidation of glucose. That is 30 ATP per glucose, complete breakdown or complete oxidation, right? So if they ask you that aerobic glycolysis gives how many, you will say 7. But if they ask you complete oxidation of glucose, then you will say 32. So be careful what they are asking. Now compare these two enzymes. One is alpha ketoglutarate dehydrogenase of TCA cycle, other is pyruvate dehydrogenase PDH complex, that is the link reaction enzyme. Both these enzymes are multi-enzyme complex. Both these enzymes require same 5 coenzymes, I already told you. But the difference is this point, that alpha ketoglutarate dehydrogenase is not regulated by phosphorylation and dephosphorylation, but PDH complex enzyme is regulated by phosphorylation and dephosphorylation. That is, insulin in fed state will cause dephosphorylation and will activate link reaction. Link reaction is activated in fed state. But on the other hand, in fasting state, link reaction will be inhibited by glucagon, which is doing phosphorylation, right? Now TCA cycle occur in aerobic state. We all know it occurs in aerobic state. But any enzyme of TCA cycle does not require oxygen. No enzyme of TCA requires oxygen. Then my question is, why TCA cannot occur in anaerobic situation? See the reason. Let's say there is a situation that oxygen is not there. When oxygen is not there, then TCA will occur because no enzyme of TCA requires oxygen. So TCA will occur. That is, first few cycles of TCA will be occurring. Now when TCA will occur, then lots of NADH and FADH2 will be produced. This NADH FADH2 has to go into ETC for giving ATP. But we have taken the situation oxygen is not there, so ETC cannot occur. So NADH and FADH2 will accumulate. NADH, FADH2 accumulate. Or we can say NAD and FAD is depleted. That is, all the NAD and FAD of the cell is converted to NADH and FADH2. So now NAD FAD depleted means TCA will stop, right? So this is the reason that TCA cannot occur in anaerobic condition. Now let's see the rate limiting steps in TCA cycle. See, I said rate limiting steps. Usually in some pathway or cycle there is only one rate limiting enzyme or step, but in case of TCA cycle there are 3 enzymes which are rate limiting, 3 steps which are rate limiting. That is a very special thing here. Actually, in different situations, the different enzymes can be rate limiting, and they are citrate synthase, alpha ketoglutarate dehydrogenase, and isocitrate dehydrogenase. These 3 enzymes can be rate limiting. Let's say in some situation citrate synthase is rate limiting. In another situation alpha ketoglutarate dehydrogenase is rate limiting. In another situation isocitrate dehydrogenase is rate limiting. That body has to decide depending upon various complex conditions, right? And now irreversible steps of TCA cycle. There are 2 irreversible. One is citrate synthase and another is alpha ketoglutarate dehydrogenase. If both are given, then go for citrate synthase, right? Now tell me, there are how many dehydrogenases in TCA cycle and there are how many kinases in TCA cycle? Dehydrogenases are 4, that is alpha ketoglutarate dehydrogenase, then isocitrate dehydrogenase, then succinate dehydrogenase, and the last one is malate dehydrogenase. These 4 dehydrogenases are there. Out of these, first 2 which I have written, they are doing oxidative decarboxylation, but the other 2 are only doing oxidation. And you know, in alpha ketoglutarate dehydrogenase and isocitrate dehydrogenase, NADH is formed. Succinate dehydrogenase will form FADH2, and malate dehydrogenase is also forming NADH, right? Now, there is only one kinase, that is succinate thiokinase in TCA cycle, which is the only step of substrate level phosphorylation. Now we have to see the carbon dioxide which are released in TCA cycle. They are from oxaloacetate or acetyl-CoA. The carbon dioxide in TCA are derived from the carbons of oxaloacetate. If simply question is asked, then you go for oxaloacetate. If oxaloacetate not given in the option, then you mark acetyl-CoA, right? Now, I am going to tell you the detail of this thing also. If you don't understand the detail, then you should cram only this much, right? But once in the video, I will be taking this detail, watch it once. If you understand, this is okay, go with the concept, otherwise cram this, okay? So, let's see the detail. What happens actually, you know, oxaloacetate is having 4 carbons. It will join with acetyl-CoA, which is 2 carbons, and the cycle will occur, many reactions will occur. There is removal of 2 carbon dioxide, and this is first cycle which I'm talking. In the end, you know, again oxaloacetate is formed, and in second cycle, again 2 carbon dioxide are removed, right? So let's say we are doing this as we are carrying TCA reactions in lab in vitro conditions, or in the lab we are having it, so we can give colors to the carbons, right? So let's say we give red color to the carbons of oxaloacetate. Let's give black color to the carbons of acetyl-CoA. Now, this carbon dioxide which is released is red in color. The oxaloacetate which we have obtained at the end of first cycle, that oxaloacetate has 2 carbons red and 2 carbons black, okay? And in the next cycle, the carbon dioxide which are removed are black in color, okay? So, if we are talking of only one cycle, simply if we are talking of one cycle, then the carbon dioxide are from the carbons of oxaloacetate, right? But if we talk of a previous cycle also, if we are talking of this carbon dioxide, then this carbon dioxide is coming from previous carbons of acetyl-CoA, right? So it depends on the question what they are asking, but if you are clear in this detail, then be it any question, you will be able to answer, okay? So whatever question you will be able to answer, just go with the concept. I revise once, that the carbon dioxide is coming from oxaloacetate from that particular cycle for which we are talking. But these 2 carbons which are coming in the form of carbon dioxide, these carbons are from the previous acetyl-CoA, right? So this way you can solve the questions, right? So this is it with this video. Thanks for watching.